Thank you for the opportunity. Thank you, the United Nations and FAO, Food and Agriculture Organization, for letting me give this presentation today. So my name is Weibin Liu, and I'm Vice President and Special Assistant to the Chairman of BGI Group. And together with my colleagues at BGI Research, I work very closely in advancing our new technology, Stereo-seq, in the field of spatial biology. The talk, as I said earlier, will be large field of view, high resolution, spatially resolved transcriptomics using DNA nanoball Panard arrays. Next slide. Okay. So cells have very important and specific spatial context. So to truly understand biology, we really have to look at the hierarchical organization of what life is made up of. So at its most basic unit, you have the cell, but there's a lot of different types of cells, and they have very different functions, and the different omics profiles within each of these cells define what kind of cell types they are and what kind of cell states they may be in, and what their functions are, and then how they're organized into tissues, and how those tissues make up organs, and how the organ systems function together in the entire organism. Is having that kind of hierarchical information at a very high resolution is important for understanding some of the most basic biological concepts like physiology, really understanding physiology, really understanding development and aging across the entire lifespan, really understanding what is normal, what is not normal, and therefore what constitutes disease, and what can you do about it. So so the field of biology has really been converging on being able to understand cellular heterogeneity, not only through single cell sequencing and understanding populations of cells, but also being able to retain their original spatial context and how that spatial context evolves over time in order to describe those processes. Next. There have been efforts made to unravel spatial complexity over the last 40 years. So some of the earliest efforts were focused on imaging-based techniques, and primarily around in situ hybridization. And then more recently, increasingly with sequencing-based techniques. And all these technologies try to tackle that challenge that I mentioned in the previous slide, which is really how do you get the spatial information and how you use that to study cellular heterogeneity in their spatial context. But there have been limitations. For example, with in situ hybridization, you have a limited number of targets that you can target, and so scalability and how much total information you can extract is limited. So with latest developments with sequencing-based technologies, some of the limitations have been around resolution and also field of view, and also whether it is whole transcriptome or whether it's only target sequencing. Next slide. And this entire trend has been very apparent in recent years. For example, in 2020, the Nature Method of the Year was spatially resolved transcriptomics, which profiled several groups working on some of the most cutting edge advances in this field. One of which was BGI Research, which at the time described some of its earlier results with our proprietary technology, Stereo-seq. Next slide. So in order to understand spatial cellular heterogeneity you would have to get down to single cell resolution. And that's really been the goal of this entire effort. So some of the previous efforts made have not been able to get down to single cellular resolution. So for example, you have some of the existing technologies like Visium. We have a spot size so when you try to do the spatially resolved sequencing, the spot size for Visium is 55 microns, which is, if you think about the average size of a cell, which is 10 microns by 10 microns, one spot covers multiple cells, covers maybe a couple of dozen cells, which means that in order to get true spatial information, there's a lot of limitations. You have to go through deconvolution technologies to try to guess what kinds of, combined with single cell sequencing data, where in space the different cells approximately are. But then with such a big spot size, and also with a lot of gaps in between the spots, when you have center resolution of 100 microns, it means that you're losing a lot of spatial data, and you're getting at best an average signal for the spatially resolved data. You're not able to get down to single cells. What's going on with the slides? Okay, thanks. So you're not able to get down to single cell resolution, and with some of these other technologies, they're also limited by the scalability in terms of the field of view and capture area. So for example, with the field of view in measured in millimeters for these different technologies, it may be useful for smaller tissues when you're trying to examine things like Drosophila embryos or zebrafish embryos. But what if you wanted to use this technology? What if you wanted to get spatially resolved single cell omics information across large organisms or large organs and tissues? You would not be able to piece these together very efficiently. So these challenges have been primarily in resolution and capture area, and all the while trying to be able to retain the in situ unbiased whole transcriptome. Next slide. So our technology that we use in our sequencers was what inspired the development of Stereo-seq. So in our massively parallel sequencing technology, DNB-seq, which stands for DNA nanoball sequencing, we use large silicon wafers with a very, very fine resolution patterned array of DNA nanoballs etched onto the wafer. So we have the nanoballs measure only 220 nanometers in size, and they're spaced 500 nanometers apart, center to center. And each of these nanoballs, there's independent sequencing reactions, all done in parallel, which results in our massively parallel sequencing. So we wondered that, and furthermore, the size of the chips is scalable up to 13 centimeters by 13 centimeters currently. So we wondered if we would be able to repurpose this technology to make a breakthrough in the area of spatial biology. Next slide. And we were successful at doing so. That's exactly what we did. So we took our DNB-seq chip, and we generate the DNA nanoballs instead with a random CID barcode of 25 bases, which gives us a unique barcode for every nanoball on this gridded patterned array across this large chip. And then we sequence all the nanoballs, and then so we get a unique 25 base sequence for each of these nanoballs. And then when we store that as a separate file, and then we attach the UMI barcode to distinguish between different molecules, and also poly-T probe in order to capture the mRNA. And then when we do this, we finish making the Stereo-seq chip. So now you have each nanoball having multiple probes that can hybridize with multiple RNAs, and each nanoball also being barcoded by a coordinate ID that corresponds to its precise location on the XY plane of the chip. So this way, once you sequence it and you get the results, you'll be able to recreate the spatial location in silico of where all the reads were. So next, for step number 4, with a completed stereo-seq chip, we then take a slice of tissue and we load it, we fix it, and then we do around nucleic acid staining in order to get an image for to see where the nuclei are for all the cells on the tissue section, in order to do image registration later together with our together with the in situ transcriptome data, and then we permeabilize it, which results in the mRNA hybridizing to the probes on the DNA nanoballs, and then we do reverse transcription to generate cDNAs for the captured mRNA, and then we wash that off, and then we prepare for sequencing, and then with the sequenced results, then we run it through our own bioinformatics pipeline to get to recreate the spatially resolved transcriptome profile. Next slide. Okay. So we again, we have very large field of view that compared to existing technologies. So when current technologies are measured in terms of just several millimeters by several millimeters, we're measuring 13 centimeters by 13 centimeters at our largest chips, which means that we'll be able to conduct these spatially resolved single cell omics experiments on very large tissues, including, for example, if you look at the monkey semi-brain section on the right, it fits very comfortably on a 7 centimeter by 7 centimeter chip. And the human brain can also fit on the largest chip. And not only in terms of like human tissues, but animal tissues, or even whole animals if they're small enough. And of course, a lot of exciting applications in the plant field as well. Next slide. So compared to existing technologies in terms of field of view and resolution, where we really blow the field away. And given that our resolution, because of the nature of our DNAB-seq chips that we built this technology on, the resolution being 500 nanometers means that for an average size cell of 10 microns by 10 microns, we get 400 spots at subcellularly for each cell, with each spot being able to capture multiple transcripts, which demonstrates the single cell or subcellular resolution of stereo-seq. Next slide. So we wanted to validate this technology after we developed it. So we first did it on the mouse olfactory bulb, where we took 2 sections and we performed stereo-seq on these sections. And then we used bin 50. We used a spatially constrained, unsupervised spatially constrained clustering analysis to basically split the spatial data into a grid measuring 25 microns by 25 microns for each of the squares in the grid, and then we do statistical analysis in order to we do clustering analysis in order to to use the grids to identify the anatomical regions within each of the sections, and then we do and then when we look at mRNA capture efficiency, we see that we capture about 1,500 transcripts for a 10 micron bin from this data, which is roughly about the area of a single size cell, and then also they corresponded to about 500 to 600 genes per 10 micron bin, and we see very good reproducibility between the 2 different sections. Next slide. And when we compare with existing technologies, the transcripts that we capture at different resolutions is far more than other technologies. So whether at 2-micron bin or 10-micron square bin or 100-micron square bin, we see that we capture significantly more transcripts than existing technologies. And then also, we want to validate the spatial localization of example genes, whether Stereo-seq accurately accurately captures where we expect to see them. So we went to the Allen Brain Atlas in situ hybridization database at the very bottom there, where they have well-documented locations for these two example genes, and we compared with our data that we got from Stereo-seq for these two olfactory bulb sections, and we see that the gene expression is localized where we would expect to see from the Allen Brain Atlas data, whereas it's you cannot say the same for some of the other technologies that you see in between there. Next slide. And then we wanted to see, so because the resolution is only 500 nanometers and we can get down subcellular resolution, so we wanted to see what kind of analysis we can do with that. So when we look at the nucleic acid stain for a mouse hemibrain section on the left there, and we look at the detect DNB image on the right, you see that by zooming in on detect DNB, the resolution is high enough that you can actually see where the transcripts are localized around where the nuclei are. So even just from the spatial location of all the transcripts you get from the Stereo-seq data, you can actually, with some of the better data sets, you can actually see where the cell boundaries are approximately based on the location of the nuclei. And that analysis becomes much more accurate when you combine it with the nucleic acid stained image. So you combine the image location of the nuclei with the DNB location of the nuclei, and then you can approximate the boundaries of the cells, so you could assign the location of all the transcripts to their associated cell for further analysis, for further clustering analysis at the single cell level to annotate individual cells. So on the lower left there, that's what we did. We first took the mouse hemibrain slice, the coronal slice here, and we did bin 50 analysis to first, breaking it down to square bins and understanding where the approximate anatomic regions are. And then within each of these anatomic regions, we then performed image-based cell segmentation as shown above in order to identify all the individual cell clusters within each of these anatomic regions, color-coded. And then you can see on the lower right that produces very fine maps of the cellular heterogeneity that you see in the different regions of this mouse hemibrain. Next slide. And we also explored how big of a factor is diffusion in this, because when we permeabilize the tissues, it opens it opens up the chance for diffusion, which may affect the spatial localization of transcripts to which cell they belong to. So we compared the location of mitochondrial transcripts versus nuclear transcripts, and we see that the distance is less than the width of a cell, average at about 6.8 microns diffusion distance, whereas compared to in situ hybridization, which is 5.3 microns, we're only a little bit worse, but we're also making significant improvements with new workflows in order to get that the same or better than in situ hybridization. Next slide. And then so next we apply this technology to unlock the black box cell organization during embryonic development. So we specifically studied the mouse embryonic development from days 9.5 to 16.5 using Stereo-seq. Next slide. And then we first did bin 50 analysis in order to look at the tissue locations for all of the different tissue types in each of these different time points for the mouse embryo. We did a total of 53 sections of the mouse embryos at the different time points, and we're able to see recreate the developmental trajectory of all the different tissues at all the different time points compared to what's currently known, very accurately recreated. Next slide. And then when we do further deeper clustering with the from the previous bin50 data in specific tissue regions, we're able to see tissue subregions. For example, with a spinal cord and with a developing mouse brain data, and we're also able to see microstructures like what you see with a kidney up there, just by doing a round of reclustering or deeper clustering. Next slide. And then we also explored the functional aspects that we can see from Stereo-seq data. So on the left there, we used hotspot. We used the algorithm hotspot to look at non-random variation in gene expression. We're able to identify gene modules associated with the different tissue types there. And also, if we apply other algorithms like SNCC, which is used for single-cell sequencing, together with hotspot, we're also able to identify the locations of the different regulons and also the modules of the regulons. Next slide. And when we apply image-based cell segmentation to segment individual cells on the day 16.5 mouse embryo, we were able to get about 300,000 cells at single cell resolution, and we're able, through cell clustering, we were able to identify all the different cell types across this tissue section, and also reclustering, for example, of epithelial cells and of chondrocytes, we're able to see the cell subtypes as well. And in the video on the left, on the right, you can see zoomed in that all the individual segmented cells. Next slide. And we also explored cell fate dynamics, specifically in dorsal midbrain development. So we looked at the dorsal midbrain, and we did cell clustering, and we looked at a group of cells circled there on the upper left. You see the radioglial cells, the RGC, are the progenitor cells to two cell types, glioblasts or neuroblasts. So there's two different cell fates from RGC. And we looked at days 12.5, 14.5, and 16.5. And we see that day 12.5, the progenitor cells are evenly spread throughout the dorsal midbrain. But by day 14.5, differentiation into the different cell types has occurred, and you can see spatially where the cell fates have gone toward glioblasts or from neuroblasts. You see progenitor cells remain mostly localized in the caudal region of the dorsal midbrain. By day 16.5, that patterning of differentiation is even more apparent. And on the right, you also see that by looking at the two different cell fates for radioglial cells, using Stereo-seq data, we're also able to see the differentially expressed genes and ligands for these different cell fates. Next slide. And last, for this particular paper, we studied, we wondered if the mouse Stereo-seq data could serve as a potential model for better understanding human developmental disorders, as humans and mice, while not the same, are somewhat close. So we went to the developmental disorder genotype to phenotype database, and we identified 1,959 SNPs from human GWAS data, and we mapped those SNPs to the mouse Stereo-seq data across all the time points, and we're able to see where all the different SNPs associated with these human developmental disorders are where and when that they're being expressed. So, for example, when we look at Robinow syndrome, which Wnt5a is implicated, Wnt5a and MSX1 are both implicated, and we see that the spatial locations of Wnt5a and its homeobox gene and MSX1 are actually, localized in the same areas where you expect to see the phenotypes for Robinow syndrome in the maxilla region and in the limb region. And at the same time, you're also able to see what cell types are expressing these. So there's 2 different cell types that are expressing it in the maxillary region, whereas only 1 cell type is expressing it in the limb region. So this gives us much richer insights into the data from GWAS and how that actually translates to the developmental story and how that leads to the disorders. Next slide. And so we applied Stereo-seq similarly to build one of the first panoramic atlases for not only the mouse development, but mouse embryonic development, but also for zebrafish development as well. Next slide. And we also did it for Drosophila embryos and larvae, and we did it with multiple additional slices stacked up so that we recreate a 3-dimensional spatial transcriptomic atlas of the Drosophila embryo and larva. Next slide. We also did this in plants. So for plants, what's interesting is plants have cell walls, which makes it difficult to do single cell sequencing because you have to break apart the cell walls to get to the individual cells. But for Stereo-seq, it's actually a big advantage because with the cell walls, by staining the cell walls, you get a very clear segmentation of where the different cells are. So much more accurate than what we're able to do with animal cells currently in terms of cell segmentation. So we actually call this single cell Stereo-seq, and we did this on the Arabidopsis, and we were able to see exactly where all the cell boundaries are and where all the transcripts are. We were able to identify a lot of interesting gradients that explain the plant physiology and function. Next slide. And of course, we have recently we also have another cover paper in Science examining the incredible regenerative capacity of the axolotl, which any part of its physiology. Hello. Okay. Yeah. So we. I'm sorry. Okay. So we did. We looked at the tremendous regenerative capacity of the axolotl, and it could even regenerate its brain. So we created lesions in the brain, and we looked at the axolotl, and we explored its regeneration over the course over 6 different time points post injury, and we compared it with. Interestingly, we found that some of a certain group of cells are induced by injury, and actually they go through a series of cell state transitions that are very similar to the axolotl embryonic neurogenesis process. So we see a lot of interesting similarities between the neurogenesis and development, and also in the process that the induced progenitor cells from the injury go through, the cell state transitions they follow in order to re-heal the brain, which is very interesting because we're able to, for the first time, really understand this incredible process from a spatial temporal single cell omics standpoint. Next slide. And of course, it has incredible applications in solid tumors as well, and in digital pathology in general. So we applied Stereo-seq to look at the tumor microenvironment. So for example, specifically on the invasive edge of solid tumors. So we looked at one type of cancer in the liver, and we were able to look at the margins, the boundaries near the edge between the tumor and the healthy tissues. And we're all able to see with great resolution where all the different immune cells are, where all the stromal cells are, and also where all the cancer cells are, and what cell types they are, and what cell states they're in. And through this kind of data, we were able to better understand how injury produced by the invasive front of the solid tumor of the healthy tissue actually creates an immunosuppressive environment that recruits macrophages there that actually helps induce, that suppresses the immune system, actually induces further growth of the tumor and results in a worse prognosis for the patient. So all this would not be possible without having what Stereo-seq enables. Next slide. So Stereo-seq is poised to really transform and revolutionize the area of spatial precision medicine. So, in the example I just mentioned with the invasive frontal solid tumors, it shows its incredible potential in really understanding the tumor microenvironment, but and being able to explain, from a spatial single cell perspective exactly how tumorigenesis occurs, how they metastasize, why do they metastasize where they do, and how do they do it, and when, and under what circumstances, and also for how do you improve therapies, how do you overcome resistance, how do you stratify different patients, how do you combine incorporate spatial transcriptomics, spatial proteomic signatures to really explain, to really more fully explain, the prognosis of the patient, what kinds of treatments will produce the better outcomes, why do certain treatments fail, and how do you overcome those failures. So this means that you look at, in the 19th century, our understanding of pathology was really cellular pathology using H&E staining and immunohistochemistry. In the 21st century, we're getting into molecular pathology, but still without full understanding. So from this year onwards, we're really getting to the new field of spatial precision medicine, where you'll be able to combine the spatial multi-omic signatures enabled by this technology, from combining imaging, DNA, RNA, and proteins to truly be able to create personalized treatments that can revolutionize our understanding of precision medicine, and all this will change the field of pathology completely. Next slide. So we contribute these results to a global call to action in forming the Spatial Temporal Omics Consortium. So the results that I just described, they were published over the last several months on the covers of Cell, Developmental Cell, and Science, just to name a few of the more prominent ones, and many more have been submitted and are in the process of publication as well. We contributed these results into forming the Spatial Temporal Omics Consortium. Next slide. The Spatial Temporal Omics Consortium is an open collaborative research initiative established to unite, organize, advance, and share the global scientific efforts in order to advance advance our understanding in very fundamental biological areas like the physiology of all living things, the development and aging, and and disease, and also really understanding where we come from in terms of evolution. So more than 90 different scientists form the charter members of this consortium, coming from almost 30 different countries. Next slide. Next slide. So right now, so STOmics, which is the subsidiary within BGI Research that is responsible for the development and of Stereo-seq and making this technology accessible, has generously provided a grant program in support of the Spatial Temporal Omics Consortium, where if you scan this QR code, you will be able to go to a place where you could apply with a study proposal how you could use this technology to further your understanding in the area that you're studying, where we will provide 12 Stereo-seq chips that's 1 centimeter by 1 centimeter each, and also 5.4 terabytes of sequencing to help support your research efforts. Next slide. And then, of course, looking forward into the future, the current technology that I just mentioned focuses all on spatial transcriptomics, but in the future we'll also make it not only compatible with more sample types, including FFPE samples. We'll also get better resolution, better capture efficiency. We'll get better single cell binning we'll get better single cell segmentation algorithms. And then also we'll make it compatible with, on the same slide, to be able to do H&D staining, to do IHC and immunofluorescence, and also do it with enabled proteomics along with transcriptomics on the same slide to get richer information from the spatially resolved single cell data. And as well as also support the development of three-dimensional atlases with 3D reconstruction of all the Stereo-seq data in order to map entire tissues, entire organs, or entire organisms. Next slide. And last but not least, of course, user experience is very important. So we're also coming out with a fully automated solution where with less than 30 minutes hands-on time and 7 days end-to-end turnaround time, you will be able to process more than 24 chips at the same time. Next slide. So thank you very much. Sorry that I went a little bit over time, and feel free to scan this QR code if you're interested to learn more about the Spatial Temporal Omics Consortium.